If this is your first time here, this isn't a standalone video. It's one chapter in a complete story, the entire history of evolution, from the first replicating molecules to you. Every organism we mention is your direct ancestor, not a cousin species, not something similar, your actual lineage, an unbroken chain connecting you to the origin of life. That's what makes this different from every other evolution content out there. Most of it is scattered fragments, dinosaurs here, early humans there, the Cambrian explosion somewhere else, all disconnected. But they're not disconnected. They're all chapters in the same story, your story, and you can't understand any link without seeing what came before it. Lungs don't make sense without swim bladders. Mammals don't make sense without reptiles. Each chapter builds on the last, and nothing appears out of nowhere. 4 billion years of your deep history told as one continuous thread. If that sounds interesting, Chapter 1 is in the top right corner. I'd recommend starting there. If you've been watching from the beginning, you already know what this is. Let's keep going. Our lineage now exists as single-celled eukaryotes, complex cells with mitochondria, nuclei, internal membrane systems, and cytoskeletons. These cells are larger, more energetically capable, and more structurally sophisticated than any prokaryote. For hundreds of millions of years, they diversify as single-celled organisms, exploring different ways to live in aquatic environments. This period sees the evolution of numerous protist lineages. The term protist simply means eukaryotes that aren't plants, animals, or fungi, and within this diversification, the specific adaptations emerge that will eventually enable multicellular life, including the lineage leading to animals. Early eukaryotes were predators. Unlike bacteria that absorb dissolved nutrients through their membranes, eukaryotes could engulf entire cells through phagocytosis. A eukaryotic cell extends protrusions of its membrane around a bacterium or smaller eukaryote, wraps completely around it, and internalizes it into a membrane-bound compartment called a food vacuole. Digestive enzymes break down the captured cell, and the nutrients are absorbed. This hunting lifestyle immediately creates a different selection pressure than those acting on absorptive bacteria. Success as a predator requires finding prey, capturing it efficiently, and avoiding becoming prey yourself. These demands drove the evolution of better sensory capabilities, proteins embedded in the membrane that detect chemical signals from potential prey or predators. They drove improvements in motility, the ability to move toward prey or away from threats, and they drove innovations in cell shape and behavior that made hunting more effective or escape more likely. Some eukaryotes evolved flagella, long whip-like appendages powered by molecular motors that propel the cell through water. Eukaryotic flagella are complex structures with a characteristic 9+2 arrangement of microtubules, 9 pairs forming a ring around 2 central singlets, all enclosed in a membrane extension from the cell surface. The microtubules slide against each other using motor proteins, creating wave-like beating motion that pushes water and moves the cell. Flagella allowed active swimming rather than drifting passively, enabling pursuit of prey and directed movement towards favorable conditions. Other eukaryotes evolved different solutions. Some developed cilia, shorter, more numerous hair-like projections covering the cell's surface that beat in coordinated waves, moving the cells or creating water currents that sweep prey towards the cell. Some became primarily sedentary, attaching to surfaces and extending sticky or adhesive projections to capture passing bacteria. Some abandoned predation entirely and involved photosynthesis, after engulfing photosynthetic bacteria in events similar to the mitochondrial merger, turning those bacteria into chloroplasts. That last point bears emphasis. Just as mitochondria arose from an archaeon engulfing a bacterium, chloroplasts arose from a eukaryote engulfing a photosynthetic bacterium. The lineages that acquired chloroplasts diverged into what we broadly call plants and various photosynthetic protists. This is not our lineage. Our ancestors remained non-photosynthetic, predatory cells. Plants and animals share a common eukaryotic ancestor from before the chloroplast acquisition, but after that point, their evolutionary paths separated. Plants solved the energy problem through photosynthesis. Our lineage continued relying on consuming other organisms. Within the predatory eukaryotes, one major group called opisthecons emerged and diversified. Opisthecons includes animals, fungi, and several single-celled lineages. The defining feature is subtle. If they have a flagellum, it's single and positioned at the rear of the cell, pushing rather than pulling. This is a mere morphological detail, but it marks a real evolutionary grouping confirmed by genetic analyses. Our lineage belongs to opisthethons, specifically to the branch within opisthethons that led to animals. The closest living single-celled relatives of animals are cyanoflagellates, small predatory cells with a distinctive morphology. A cyanoflagellate has a single flagellum surrounding at its base by a collar of fine, finger-like projections called microvilli. The flagellum beats, creating a water current that flows through the collar. Bacteria get trapped in the collar, stick to the microvilli, and are engulfed and digested. It's an efficient filter-feeding strategy that works well in bacteria-rich waters. Cyanoflagellates are important to our story for several reasons. First, they are single-celled cousins, the closest thing to what our pre-animal ancestors looked like. Second, they already possess some of the molecular tools animals later use for multicellularity. Cyanoflagellates have genes encoding proteins for cell adhesion, making cells stick together, and for cell signaling, allowing cells to communicate chemically. In cyanoflagellates, these proteins serve other functions or remain unused, but their presence indicates the molecular toolkit for multicellularity existed before animals evolved. Third, some cyanoflagellates form colonies. After a cell divides, the daughter cells sometimes stay attached rather than separating. A colony might contain a few cells or dozens, all genetically identical, all performing the same function or filter feeding, but physically connected. These colonies aren't multicellular organisms in the full sense. There's no specialization, no coordination, beyond physical attachment, but they demonstrate that staying together after division is possible and sometimes advantageous. Why would staying together benefit cells? Several advantages emerge even in simple colonies. A larger structure sinks more slowly than individual cells, staying suspended in the nutrient-rich upper water layers longer. A colony can create stronger water currents, pulling in prey from a larger area. A colony is harder for some predators to consume, too large to engulf whole, requiring predators to break it apart, which takes time and effort. These modest benefits can be enough for selection to favor cells that remain attached over cells that separate. The transition to true multicellularity requires more than just cells sticking together. It requires specialization, different cells taking on different roles. Specialization emerges through differential gene expression. All cells in a multicellular organism carry the same genome, but different cells activate different subsets of genes, producing different proteins and therefore different structures and functions. The mechanisms enabling differential gene expression existed in single-celled eukaryotes long before multicellularity. Even a solitary cell needs to regulate which genes are active in response to environmental conditions, turning on genes for digesting nutrients when food is available, activating stress response genes when conditions deteriorate, expressing different genes at different stages of the cell cycle. The regulatory networks controlling gene expressions were already sophisticated in single-cell ancestors. What changed in early multicellular organisms was using these regulatory systems in a spatial context. Cells in different positions within a colony experience different conditions. Cells on the outside face the external environment directly. Cells on the inside are shielded. Cells at the front of a moving colony encounter fresh water. Cells at the rear experience water already filtered by those in front. These differences in position create differences in signaling molecules the cells encounter, which trigger different regulatory responses, activating different genes. Simple spatial patterns emerge automatically. In a spherical colony, outer cells might activate genes in response to detecting unfiltered water, while inner cells, not detecting that signal, leave those genes inactive. If outer cells then produce a signaling molecule that diffuse inwards, inner cells respond by activating different genes. Within a few cell divisions, the colony now has two cell types, outer cells expressing one set of genes and inner cells expressing a different set. This is the beginning of differentiation. Early differentiation was probably subtle and not particularly beneficial, but occasionally having different cell types provided advantages. Perhaps outer cells specialized in filter feeding while inner cells specialized in reproduction, protecting the next generation from environmental damage. Or perhaps some cells specialized in adhesion, producing extra sticky proteins that held the colony together more reliably. Any functional specialization that improved colony survival or reproduction would be favored by selection, gradually increasing the degree and complexity of differentiation. The evolution of extracellular matrix, materials that cells secrete around themselves, enhanced multicellularity substantially. Single-celled eukaryotes often secrete glycoproteins and other molecules that coat their surfaces, providing protection or helping them stick to substrates. Multicellular organisms elaborated these secretions into structured materials, filtering the spaces between cells. The extracellular matrix provided mechanical support, holding cells in specific geometric arrangements, and also served as a medium for diffusing signals between cells. Cell signaling systems became more sophisticated in multicellular contexts. Single cells already had receptors on their surfaces that detected external chemicals and triggered internal responses. In multicellular organisms, cells use similar receptors to detect signals from other cells in the same organism. A cell can release a signaling molecule that binds to receptors on nearby cells, triggering those cells to change their behavior, perhaps dividing or differentiating into a particular type or moving to a different location. These signaling systems allow coordination. Cells in different parts of a colony could respond to each other's states and adjust their own behavior accordingly. If cells at one end detected abundant food, they might signal cells at the other to stop dividing and focus on feeding. If cells in the center detected damage, they might signal outer cells to increase production of protective molecules. Coordination transformed a colony from a loose aggregate into a functional unit behaving coherently. The oxygen-rich environment, created by billions of years of cyanobacterial photosynthesis, played a crucial enabling role. Higher oxygen levels supported higher metabolic rates and allowed organisms to grow larger without their interiors suffocating. Early multicellular organisms were probably small, just dozens or hundreds of cells, but oxygen availability meant there was no fundamental barrier to growing larger. Oxygen diffuses through tissues, and organisms can remain aerobic as long as no cell is too far from an oxygen source. This becomes a constraint only at much larger sizes, eventually requiring circulatory systems. But for early simple multicellular organisms, ambient oxygen diffusion was sufficient. By about 800 million years ago, our lineage existed as small multicellular organisms, probably only a few hundred cells large, living in marine environments. These organisms had differentiated cell types, not many, perhaps only 2 or 3 distinct types, but enough to show functional specialization. They had extracellular matrix holding cells in defined arrangements. They had cell signaling systems allowing coordination. They could grow and reproduce, with some cells specializing in reproduction, while others specialized in feeding or structure. These weren't animals yet. They lacked tissues, organs, and the sophisticated organization that defines animals. But they had crossed the threshold from single-celled to multicellular. Every subsequent innovation in our lineage builds on this foundation. Groups of cells working together, specializing into different types, coordinating through signaling, and organized structurally through extracellular matrix. The path from here to recognizable animals involves elaborating these capabilities, adding more cell types, organizing those types into tissues, coordinating tissues into organs, and developing the specialized structures like nervous systems and muscles that allow complex behavior. That elaboration and the first true animals emerge in the next chapter.